Sugars can be shown in two different forms. The first is a linear or line model, and the second is as a ring structure. You'll mostly see ring structures in our text, but it's good to be familiar with what the line structure looks like. As our friend Hank told us, there are two types of carbohydrates, simple sugars and polysaccharides. Within the polysaccharides, we have two groups, storage and structural. Storage polysaccharides, such as starch or glycogen, have a primary role to store the chemical energy of these molecules. During photosynthesis, light energy is harvested and turned into chemical energy through the production of sugar. These sugars are stored in plants as starch. This is how we get potatoes and carrots, etc. Plants can also produce structural carbohydrates. These structural carbohydrates provide the support for the plant as cellulose. This is how trees can become as tall as they are. The differences between a structural or a storage carbohydrate comes from the glycosidic linkage. The alpha linkage, shown here on the left, gives the carbohydrates the chain/branch structure, while the beta, where every other glucose molecule is flipped, as you can see here on the right, allows the carbohydrates to form thread-like structures that provide support. The alpha linkage can be broken down and metabolized by our bodies, while the beta linkage cannot. That's why we can't live on grass. Here you can see the structural differences between storage and structural polysaccharides. Take a moment and sketch the differences between these two polysaccharides. Make sure to label them and include some examples and bring them to class for points in discussion. As you can see here on the left, we have plant-based storage polysaccharides on the top and animal-based storage polysaccharides at the bottom. Now let's test our knowledge. If actively growing cells are fed 14 carbon labeled glucose, which basically means we've radioactively tagged the glucose so we can detect it on imagers, what macromolecules will become radioactive first? Pause the recording and think about this. Where have we been talking about glucose so far? Good job! Starch is a carbohydrate, hence the reason why we would see this show up first. Now let's look at our next large biological molecule, lipids. Lipids are not considered macromolecules because they are not very large and do not have a true monomer like the other molecule classes. Hank has already showed us the difference in the two types of fats with his peanut butter example. In this picture, you can see how the saturated fats chemical structure helps them remain solid at room temperature, and while the kink in unsaturated fats stays liquid at room temperature. Hank also mentioned a different lipid that is important to us. This is a phospholipid. It is named so because of a combination of the lipid at the tail and the phosphate group at the head. The phosphate head is hydrophilic, meaning water-loving, and the lipid end is hydrophobic, meaning water-hating. So when these combine together, they make a plasma membrane made up of phospholipids. The plasma membrane borders the edges of all cells and maintains a balance of nutrients and waste within the cell. Other membranes that border organelles are also made up of these phospholipids. One last group of lipids are steroids. Steroids can serve as sex hormones or even as cholesterol. Examples of these hormones are testosterone and estrogen. We'll discuss proteins next and their role in hormones, but it is important to note that lipids can also serve as hormones. Let's test our knowledge on fats. Which of the following statements concerning saturated fats is true? Pause the recording and think about your answer. Once you have chosen an answer, push play to see if you are correct. Good job. The answer is B. Our third molecule is proteins. To start us off, let's look at how proteins are folded.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure. In most proteins, Parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself. Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups. Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
Here is an opportunity to practice one of our chapter objectives from Chapter 1. Which theme in Chapter 1 can be applied to our example of sickle cell? Write it down and bring it to class for points in discussion. Sickle cell shows us what happens when proteins are not folded correctly. The protein function is tied directly to the shape of the blood cell. And in sickle cell, our normal donut-shaped red blood cells turn into sickle-shaped blood cells that are unable to carry oxygen to tissues. We will discuss sickle cell more in depth in class, but for the moment, think about how a disease that inhibits red blood cells from carrying oxygen to the tissues, as we know is needed in all biological functions, to still be a persistent disease in our culture. Why has this disease been allowed to succeed and continue being passed on from generation to generation? What reasons could we see for this? Protein folding can be undone by a variety of factors, including heat and pH. It is why our body homeostasis is so important. Any changes outside our biological range can cause our proteins to unfold, and once unfolded, proteins cannot refold. Proteins have 8 main roles in the body. Let's take a look at each of these. The first role is as an enzyme.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
The second role is as a defensive protein, such as an antibody.
The immune system makes defensive proteins called antibodies that bind to invaders, such as the virus shown here, and mark the foreign objects for destruction.
Thirdly, protein can act as a storage protein. Similar to a storage carbohydrate, these proteins store amino acids, especially those we cannot make on our own.
Storage proteins stockpile building components that cells can use to make other proteins. Storage proteins in seeds provide raw materials used by the developing plant, unless an animal eats them first. Ovalbumin, the main substance in egg white, serves as a storage protein for developing chick embryos.
Transport proteins help move molecules in and out of the cells.
Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell.
We've already discussed hormones in this lecture. Proteins can also serve as hormones.
Signal proteins include hormonal proteins that help coordinate an organism's activities by acting as signals between cells. For example, insulin, a hormonal protein secreted by the pancreas, signals in animal cells to take in and use sugar. The hormone receptor is also a protein.
If you've seen a commercial for a medication, you've most likely seen an example of our next protein, receptor proteins.
Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins.
It is important to help our cells move, such as sperm, or move molecules around our cells. Contractile or motor proteins perform this function.
Contractile proteins, like the motor protein shown here, can move parts of a cell. Other contractile proteins, working together in muscle cells, can move a whole animal.
We are almost done. Lastly, just like carbohydrates, proteins help provide structural support, especially in animal cells that don't produce cellulose.
Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins.
Now let's check our knowledge. Which of these contain amino acids? So this question you have to think about two different things. What macromolecule is made up of amino acids? Which of these are not an example of that macromolecule? Pause the recording, and when you're ready, push play to find out the answer. Good job. The answer is B. Cholesterol is a lipid, not a protein, and amino acids are the monomers of protein. We are almost done. Our last macromolecule is nucleic acids. They come in two types, DNA and RNA. Let's take a look at their structure.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of two polynucleotide chains. The two DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
DNA and RNA play an important role in protein synthesis. They essentially provide the blueprints for these proteins to be constructed. While it is not important for you to know this picture now, it is best to review it so you are prepared when we encounter again in Chapter 17. For now, take a moment and list the differences between RNA and DNA and bring these to class. One last knowledge check. Human and mice differ because, once you have your answer, push play and continue on with the lecture. Good job! The answer is D. Let's wrap up by looking at our objectives for the chapter again. Hopefully you are getting closer to meeting these objectives. Make sure that you write down which objective you are still struggling with the most and bring that to class for your muddiest point. Lastly, to help you reach these objectives, here are some supplemental materials.